afternoon everyone. Sorry for the delay due to some technical issue. The session is delayed. Okay. So good afternoon everyone. My name is Nitish and I am a consultant trainer at Sankhyana consultancy service from the last 5 years. So today in this session we are going to discuss about the roles and responsibility of clinical SAS programmer. So as you know, let us first discuss about the SAS. So as you know, SAS stands for Statistical Analysis System. Right now it is used as the industry standard for the data management and analytics for the large volume of the data. It is a very flexible platform that provides the user for the various means to manipulate, analyze, process and report on the data. It also provides you the insight into the functional character of your clinical trial, that is how to create, how to analyze and make the report that is defined in your support of real-time data. So it provides the ability to write the SAS program, you can say import and export the data and converting it for presenting the analysis on it. So here the SAS play an important role starting from determining the clinical study to till the regulatory submission. So SAS clinical trial program play an essential role in the clinical trial data analysis. So as a market leader in clinical research analytics, SAS provides a secure analytic foundation and a scalable framework for the clinical analysis and submission. And it also provides the leading platform for the data transparency and allow you to securely share the historical data with the third-party researchers for the betterment of the medicine. So in this session we are going to discuss about the what is SAS, about the clinical research, what is clinical trial, what are the different phases of your clinical trial, importance of SAS in clinical research, and in the last we will also discuss about the role of clinical SAS programmer and advantage of SAS. So let us first discuss about the SAS. What is SAS? So as I said, SAS stands for Statistical Analysis System, which was developed by the Dr. James Goodnight at North Carolina State University in 1926. Now it is owned and maintained by SAS Institute. The intention of identifying this software in 1920 is mainly for the agricultural field data analysis. Nowadays it is widely used in your clinical, financial, banking industry, manufacturing industry, customer relationship management also. So all the industry have a tons and tons of data, right? As you know, so to handle, so it becomes difficult to handle such type of data, right? And analyze the data. So SAS provides you the platform to analyze and manage the data and present that data into the meaningful graphs and report. So it is your integrated software suite that is used for the data analytics, business intelligence, predictive analytics and data management. Getting? Okay. So SAS is your fourth generation language and it can easily run on all the operating systems like Windows, Unix and Linux. So how does SAS work? Okay. So first these are the this is your framework of SAS. So it first accesses the data. After accessing the data, we try to manage the data, or you can say manipulate the data. After manipulation, we analyze the data and then we present the data. So what does this access the data mean? So access the data means SAS allows you to access the data in any desired format that we want. So we can access the data that is stored anywhere, whether that data is in your file or your data is stored in another database system like Oracle and Teradata. So it can access also your CSV file, Excel file, right? And SAS help you to access data in a very easy manner. Now next we have to manage the data. So manage the data means SAS help you to manipulate the data. and it helps us to format your data into the SAS environment, and here we can create and delete the variable, and even though we can also perform the conditional processing of your data. Fine. Now next we have analyzed the data. So after the managing process, we will analyze the data to perform the simple evaluation. So here we can create the different types of report with the help of frequency, mean, procedure, like that. So we can create the different types of report like frequency count, cross tabulation report, right? So, and even though we can also create here the chart and graph plot. Okay. Now next we have the presenting the data. So SAS help you to present the data in a different form of list, graphics, reports, and summary, and it also help you to present your result in a result of analysis in a significant report in multiple formats such as HTML, RTF, and PDF. Getting? Now let us discuss about the clinical research. What is the clinical research? So clinical research is your study of health and illness in the people, and it is the way where we learn how to prevent, diagnose, and treat the illness. So clinical research describes the many different elements of scientific investigation, and it also involves the human participant and help to translate the basic research into the new treatment and information to benefit the patient. Correct? Okay. Now it also focuses on improving the knowledge of the disease, you can say developing the diagnostics method and the new treatment or the medical device to ensure the better patient care. So it is a very framed and respect precise study protocol, and it is only realized under the certain conditions. It must have the goal of increasing the medical knowledge and to be carried out by the competent person. So here the clinical research also focus on your all the necessary measures to protect those who lend themselves to the research and obtain the regulatory approval and take all the necessary legal and ethical steps, and also help you to collect the consent of those who involved in the research. So here we have the two types of clinical study, that is one is your observational study, and the second one we have the interventional study, or you can say it as clinical trial. Fine. So what happened in the observational study? So they improved the knowledge of the disease and its evaluation over the time, and they are carried out within the framework of the follow-up of the patient in the reference center, and they also contain the natural history study. Next we have the interventional study or clinical trial. So this provides the scientific evidence of the efficacy and the safety of the new drug, and you can say the new cure of the device or the new management of the context of an illness, right? So it is necessary step for the new molecule to become a drug, or you can say the new medical device to be marketed. Now we have to discuss the clinical trial here. So in a clinical trial, as you know, the participant receive the specific intervention according to a research plan or the protocol that is created by the investigator. So these intervention may be medical products such as drugs or device or some procedure which may change it to the participant behavior such as diet. So here the clinical trial may compare a new medical approach to the standard one that is already available to a placebo which contains the no active ingredient, right? So some clinical trials compare the intervention that are already available to each other. So when a new product or approach is being studied, it is not usually known whether will be helpful, harmful, or no different than the available alternative. So the investigator try to determine the safety and efficacy of the intervention by measuring the certain outcome in the participant. For example, we can say the investigator may give the drug or the treatment to participant who have the high blood pressure to see whether their blood pressure decreases or not. So here the clinical trial used in the drug development are sometimes described by the phase, and these phases are defined by the Food and Drugs Administration. Fine. So what are the different phases? So Now, so phase of your clinical trial investigate the health intervention and to collect the sufficient evidence to declare that drug is safe and efficacious. Here the process starts with the drug design and the discovery followed by the animal testing and then we proceed to the human trial. The clinical trial initiates with the safety test followed by the studies in human subject. So phase 0 or you can say the pre-clinical trial are used to determine the pharmacokinetics and the bioavailability of the drugs in animal. So initially preclinical trial studies are conducted in the animal with the different dosage of investigational drug to obtain the safety, efficacy and pharmacokinetics information. So what happened in your phase 1? Okay. So the investigator moved to phase 1 after the positive result outcome of your phase 0, that is your preclinical trial. So they used the large group of the people, you can say 20 to 50 with the no underlying health condition. And this phase run up with the several months spending more time. And in this phase a large dose of drug is used, basically the largest dose a human being can take without the serious side effect. So here after the administering of the drug, right, the close monitoring of the participant and you can say continuous reporting and documenting their body reaction is done in this phase. So in this phase here the investigator decides the route of the drugs administration, right, based on which can be the more effective and depending on the type of infection or the disease of investigation. So according to the Foods and Drugs Administration, if the medication is from the trusted source and approximately 20% of is successful, then it can then move to the next phase, that is the phase 2. So in your phase 2, right, so here this phase study test treatment that have been found to be the safe in the phase 1, but now need a larger group of the human subjects to monitor for any adverse effect. So it involved slightly more than 100 people sorry, who have the condition of the disease under the investigation, and here the dosage of the treatment is same as one that is given in your phase 1. Okay. So here the population investigated for the trial is still not enough for demonstrating the overall safety of the medication, but the data collected in this phase broaden the prospect of your phase 3. So according to the FDA, if the 30% of the drugs found to be safe in this phase 2, right, so they can move to the phase 3. Then let us see what happened in the phase 3. So here the studies are conducted even on the large population and in the different region and the country, and this is the phase right before the new treatment is approved, and it involves your 100 to you can say 300 participants with the condition the medication it treatment to treat. So this phase trial can last for the long period because it involves a large group of participants in the different regions, and this phase trial aim at evaluating how the new medication works in comparison to the other existing drug treating the same condition, and therefore here the investigator must demonstrate the efficacy, safety and effectiveness of the medication against that is of the existing drug. So here the randomized studies are used where the participants are randomly selected to form the different set or group where one set receives the new medication while the other set of the participants called the control placebo group is administered with an initial drug normally called the sugar pill, or we can also say it as a fake drug. Okay, so this phase 3 trial are usually double blind, which means that neither the participant nor the investigator know which medication the participant is taking, and this helps in your eliminating the bias during your result interpretation. Right now, in your phase 4, what happens? So after the drug is approved by the regulator, that is your Foods and Drugs Administration, Here there will still be the unanswered question about the drug effectiveness or the safety profile. So phase 4 study may be conducted to answer this question even after the medicine has been approved by the regulator and it is available by the prescription. Okay. Now, what is the importance of SAS in your clinical research? So, FDA defines the clinical trial as the voluntary research study that is conducted in the people that are designed to answer some specific question about the safety and effectiveness of the drugs, vaccine, or you can say therapies, or the new way of using the existing treatment. So, here the data generated is the most important and confidential to the clinical trial project as the fate of the drug, whether or not the drug will reach to the pharmacy shelf. It depends upon it. So here the clinical trial is conducted in the multiple center and a large amount of data is generated, which can be difficult to manage. So once the data is cleaned, right, the clinical study report should be written for submitting to the regulatory authority or for the product approval. So here the SAS, that is your biostatistical tool that is used to manage and generate the table, listing and graph for the clinical study report. Fine. And your SAS is your multi-domain application that is used to generate report, table, listing, or even you can say 2D and 3D graph chart. And the major domain of the SAS are clinical, means we can use it in the clinical industry, finance industry, banking industry, or even we can use it for the advanced analytics. In your clinical domain, fine, SAS play a major role in the data analysis and in preparing the clinical study report. So the core function of SAS in the clinical trial include your protocol design, you can say, and the patient and investigator recruitment, clinical trial management, clinical data management, data analysis, clinical supplies, regulatory and safety compliance, and the regulatory submission to the FDA. So here the SAS play a major role starting from defining the clinical study till the regulatory submission. And SAS can be the integrated with your clinical trial management system like Oracle Clinical. Fine. So here the data integration process starts with the extraction of the data, that is your data importing. So once the data is imported into your SAS environment, now then data is filtered and cleaning takes place. So once data is cleaned here, you can say error free, the SAS table are generated for the analysis of the data. And here the clinical study report is compiled using the data generated in the SAS environment. Okay. Now, so we can use it for your clinical trial modernization. What are the importance of your SAS and clinical research? So one way we can say it as the clinical trial modernization. So it helps you to adopt an end-to-end clinical analytic foundation so that we can spend more time on the data exploration, data quality monitoring, you can say, and executing the advanced analytic and statistics. And it helps you to integrate your clinical ecosystem and collaborate with the partner. And it uses the real world data to derive the insight into the clinical development process. Second importance of your SAS and clinical research is your clinical trial operational analytics. So here we use the advanced analytics to mimic the behavior of your enrollment process quickly and predict likely to outcome and test the different approach using the what if scenario. So it helps you to compare the realized enrollment versus situation and adopt the plan to stay on the target. And it also informs site supply more efficiently using the enrollment prediction. It uses the real world data to inform the data operation process and make the better decision. Third, we have the third importance of SAS in the clinical research is that clinical data transparency. So here we use the industry leading clinical data transparency to share the clinical research with the external researchers for secondary analysis and advancement of the new discovery. So here we give the researcher access to the unparalleled breadth of the available data from more than a sponsor. And it also empowers the researcher with the maximum analytical depth and flexibility that include the use of open source and the third party licensed software. and it also gives the researcher to free access to the data and allow for the transparency, platform interoperability. Okay. Now, in this slide we are going to discuss about the role of clinical SAS programmer. Okay, so clinical SAS programmer, as you know, they work in the healthcare industry where they use their programming skill to create and maintain the software that is used by the doctor, nurses, and the other medical professionals. So here the clinical SAS programmer work closely with your clinical informatics who are responsible for creating, maintaining the clinical information system, and they also work with the clinical data analyst who analyze the patient data to help the doctor and make more informed decision about the treatment plan. So as you know here, the clinical SAS programmer work as the part of the multi-dimensional project team, having only focus on delivering the quality and the timely deliverable for the variety of pharmaceutical and biotechnology clients. So here the clinical SAS programmer build the appropriate program in order to create the SAS data set from your clinical database. Okay, and the external data source and the other data source that is outlined in the clinical study protocol or statistical plan. So here the clinical SAS programmer develop the SAS macro template and you can say utility for the data cleaning and reporting. And also here the clinical SAS programmer has the working knowledge of SDTM guideline and the requirement that is comfortable in communicating with all the level of the internal team and external sponsor. So what are the responsibility of this clinical SAS programmer? So as you know, when clinical trial is conducted, a lot of data is generated during the clinical trial, and this raw data is converted into the tabular format as per the standard of CDISC SDTM guideline. So after the conversion of your raw data into the tabular format, right? So here that process is called the SDTM, that is your study data tabulation model. So after this process, the SDTM data is used for the analysis purpose, and this process is called the add-on. So after the analysis of the data set, we convert it into the TLF, that is your table listing figure. So the roles and responsibility of your clinical SAS programmer to review the CRF is to ensure the consistency with the protocol and adequacy to collect the data to meet the objective defined in the statistical section of your protocol, and also help in reviewing the CRF annotation and the database data specification. So here the SAS program also help to create the derived analysis data set and execute the analysis specified in the protocol or statistical analysis plan under the guidance of the project statistician. So here they also work closely with the biostatistician and the data management manager on the various clinical projects. They also help you to interface with the data management and the clinical to identify and program edit check for the data validation and the data management plan or study management report using the SAS. And they also help you to write the SAS program to generate the SAS table, listing and figure and the analysis data set and perform the validation of your program analysis data set, table, listing and figure. And they also perform the analysis defined in the statistical analysis plan. Okay, so these are the roles and responsibility of the clinical SAS programmer. Now in the last we will discuss about the advantage of SAS. So as you know, SAS is very easy to learn. So SAS is a very easy to learn syntax and it can be learned without any programming skill so that anyone can learn it. So here the coding of SAS are simple English statement which we use it in the day-to-day activity. Now next we have the ability to handle the large data set. So here the SAS is strongly capable of handling the large data set very easily. Third, we have easy to debug. Okay, so SAS is very comprehensible language, and here the process of debugging is very easy, and it is easy to understand and correct the errors because here the log window states it clearly. Fourth, we have the tested algorithm. So here the algorithm implemented in the SAS program is fully tested and analyzed by the developer. And every version of your SaaS is tested and in a control environment. Okay, before it release. So here the testing is possible because the SAS is closed source language. Now next we have SAS customer support. So here the organization of the SAS maintain the proper monitoring as the complete organization to analyze the SAS. Fine. So it also support it has the customer support and the SAS customer support handle all kinds of the problem. Next we have the data security. Fine. So SAS is the primary tool for many large scale companies, and here the company data is confidential. That's why it is the closed source language. So here the data in your SAS is completely secure, and in case of the office use, right, without the license we cannot extract the data. So here the manipulation is not possible because of the data security. Okay, and this is the reason behind your popularity in the corporate world. Next we have the SAS graphical user interface. So as you know, SAS is the language that has made the statistical computing easier for the non-programming users. Fine, and it has an amazing graphical user interface. And here the user interface has the various tools, like you can say you can plot, graph, and it is having the highly versatile library. Okay, so these all are the advantage of your SAS. Now so these are the branch office of Sankhyana Consultancy Service. As for here the head office of Sankhyana Consultancy Service is Kamnahalli, Bangalore, and this is your contact number. Now Sankhyana Consultancy Service have the two branch office. One is in HSR Layout, Bangalore, and another branch office is in Rohtas, Dairy Onson, Rohtas, Bihar, and these are the contact number. So if you have any technical and non-technical query, right, so you can contact at this number. Fine. So thank you for joining this session, everyone. And once again, I'm sorry for start this session late. Okay. So thank you, everyone. Thank you for participating in this session. Okay.